including on this call, and we just completed analyzing the first samples from COVID-19 patients across the course of disease, and we're designing the post-COVID-19 respiratory cohort. Next. Next, please. When we are thinking about collaborations in COVID-19, we want to think about the course of the disease. Well, all of you are familiar as those two big Gaussians. The red one is the symptomatic disease, in which around 20% of the patients will get critical to severe disease, 80% moderate. And then there is the whole purple Gaussian, which is people that are asymptomatic. And our usual assumption is that people will get either full recovery or death. What we're blind to is the post-disease sequela. Next. And this is sort of this yellow group, which is, we know that all patients that have ARDS or ICU or are in the ICU are going to have physical impairment, cognitive impairment, and mental health. Which specifically with COVID-19 and with coronaviruses, we know that there is fibrotic bands and fibrosis, but we don't know whether we'll have other sequelae like airway, vascular, and heart disease. So this is an important topic for both description, understanding mechanisms, and drug development. So when I look at collaboration interests and opportunities with industry, I look at both approaches to lung protection. How do we protect the lung using drugs and therapies? We look at enhanced recovery. How do we help the alveolar, epithelial, endothelial, capillary barrier recover? And we look at reversal of fibrosis. And the reason that I say reversal and not halting is because this is acute fibrosis and acute scar forming. And there's a good chance that many of the approaches we think of fibrosis are, could actually reverse fibrosis in these patients. Thank you.
Thank you very much, Naftali. That was great. And I'd like to introduce our next speaker. And Carrie, if you could focus, as the other ones have, on how it is that your background led you to be prepared for this moment with COVID research, your core expertise, and the questions that you're asking in the current pandemic.
Wonderful. Thank you very much. It's a pleasure to be here. So our core expertise that we've invested in as a lab at Yale over the last few years is to focus on human immunology. So we're part of the human and translational immunology section in the immunobiology department. And our angle is to study rare inherited immune disorders. So primarily pediatric patients. And we do this looking at sequencing and very mechanistic studies to really try to not only understand ways in which we can help the patients, but also fundamental biology that we can learn from these individuals, these sorts of experiments of nature that we then take into mouse models and really dissect. So having invested in this pipeline and program for other rare cases of immune disorders in children, we were poised and ready to try to contribute to the efforts to understand pediatric COVID-19. Next, please. So what we're doing for this effort is sort of diagram for you here. So there are cases at the rare extremes, and in our case, what we're focusing on primarily is on the right side, this rare idiopathic severe disease in the young. So those who you would not expect to develop severe disease, who they don't have comorbidities, but they do. They either get severely ill with respiratory disease, or more recently, a major focus for us in our lab is on this newly described multisystem inflammatory disease in kids. So by looking at these rare hypothesis that perhaps the susceptibility relates to a monogenic defect. And we can uncover those monogenic defects using unbiased whole exome sequencing in a sort of family by family basis. So we're recruiting these patients and doing whole exome sequencing on them and their parents in order to try to find that needle in a haystack, the single gene mutation that might make them susceptible to severe disease. And in doing so, we think we'll learn several things that's sort of more broadly applicable to COVID-19 generally. So the children who are highly susceptible to disease may have mutations in host defense genes or tissue tolerance genes that would be informative to understand COVID-19 pathology generally. And another effort here in the top right, the rare unexplained resistance to infection, is another angle where we think human genetics can be helpful. So those rare cases of perhaps elderly people with in nursing homes or with other comorbidities that you'd expect to get severely ill who don't. And that might, for example, point out mutations one might predict in ACE2 or other genes that are important for viral entry or infection. Next, please. So in order to really maximize impact of this, we do locally at Yale the genetic analyses together with collaborators in the Pediatric Genomics Discovery Program that I list there in the top right. But again, these are rare cases. So how do you maximize impact in the case of rare disease? And so we've joined up with a worldwide initiative called the Human COVID Genetic Effort. Next, please. And this is spearheaded by Jean-Laurent Casanova at Rockefeller and Helen Su at NIID NIH. This is just showing for you a map of all the sites that are contributing to this effort. So there are many participating sites all over the world, and they refer patients, again, young, otherwise healthy individuals who get severe disease. They refer those patients to the sequencing hubs. We are one of these sequencing hubs. You see there on the top left is the Yale School of Medicine. And then we'll share the data. We set it up so that the whole consortium can deposit whole exome sequencing, and these rare cases can then be analyzed in aggregate to really look for shared genes or shared pathways that will illuminate for us the biology of COVID-19. Next, please. But I mentioned this newly emerging pediatric multisystem inflammatory syndrome. It's now called MIS-C. It's had previous names before, but this is something within the last 2 months that has garnered a lot of interest. It's a peculiar finding, a different presentation for COVID-19 in which young people, children and teenagers, present in the clinic with severe illness. So they usually often will show up in the PICU with persistent fever, abdominal pain. It's sometimes thought that they have appendicitis, but it turns out it's not appendicitis. They have rash, multi-organ involvement with distributed shock, but not hypoxia. So quite different from the severe phenotypes in the elderly or adults. What's been found is that these children, the surge of kids in the PICU that have shown up with this phenotype is in many places in the world, it's showing up, and it's showing up roughly 4 to 6 weeks after the peak of total COVID-19 cases. The majority of these kids, they don't test positive by PCR for the virus when they show up in the PICU, but the majority of them do have antibodies against SARS-CoV-2. So this raises the hypothesis that a prior exposure roughly a month before set them up for this inflammatory syndrome that is still a mystery and something that people are actively studying. So one of the major questions we're tackling to understand this immunopathology is trying to look at whether there are signatures of pathogenic adaptive immune responses. Again, with that lag of 4 to 6 weeks, it suggests adaptive immunity. And there are parallels. This is often called atypical Kawasaki disease as well, since there are similarities with the rash. And that's also had previously been shown to be related to the adaptive immune system, and antibodies that were triggered in response to an infectious trigger then lead to this immunopathology. So this is something we're interested in studying more. Next, please. And these are the ways in which we're doing it. And partnerships with industry here, I think, would be extremely valuable. There are many high throughput screening assays that industry has to offer, and some of them under clear conditions that would really accelerate this kind of research. So at the top, you see, that's what I mentioned on the prior slides, that one of the pieces of our effort is to try to find genetic susceptibility alleles. But beyond that, we're really diving into the human immunology. And so we're taking advantage of serum samples that we get from these children, particularly when they're acutely ill, but we're also, as folks in the biorepository that Dr. Iwasaki mentioned are doing as well, longitudinal analyses in a highly collaborative team effort here at Yale, to analyze serum and look broadly in an unbiased way at thousands of serum proteins, as well as antibodies, looking both against viruses, SARS-CoV-2, other coronaviruses included, as well as autoantibody screening. So if there really is this cross-reactivity, we'd like to use unbiased ways and high-throughput assays to pull out what human antigens these antibodies might react against, and working collaboratively with others to test that Kawasaki idea about endothelial cell destruction directly by these antibodies or factors in the serum. And their cells we're taking advantage of as well. So doing multi-dimensional single cell immune profiling. So site-seq analyses that will really tell us which cell types and gene signatures are most relevant for this pediatric syndrome. And working also on T-cell assays. So things we can do in vitro to understand if the contribution of T-cells as part of the adaptive immune system. Thank you very much.
Thank you very much, Carrie. This is Kathy Lynch again. Let me please introduce my colleague, Andrew Wang. Andrew, if you could please turn on your video and audio and present your slides.
Hi, I'm Andrew. Delighted to be here. I'm a practicing rheumatologist. I see primarily patients with systemic inflammatory diseases. I have a particular interest with macrophage activation syndrome. And in my lab, one of the main questions we're trying to tackle is kind of the flip side of what Carrie just so beautifully presented, which is to try to understand non-genetic determinants of disease trajectories. So put simply, we're trying to understand why when you have two very similar people, and I'll give you an extreme example of that in a second, and they both get an insult, for example, SARS-CoV-2, why one of them may have cold-like symptoms and the other one may end up in the intensive care unit. And so extreme examples of this would be found in twin pairs. So these are genetically identical individuals. I'm showing this in a table on the left. So if you take, for example, twin pairs in systemic inflammatory diseases like lupus, in only about 50% of the time at most, you'll find that in the twin pairs you're looking at, both members of the twins have lupus. And this is true also in the panel below in sepsis, for example, looking at neonatal sepsis in monozygotic twins. And so as a clinician, I always say, well, this is the environment. And what we're trying to figure out is, trying to deconvolute the dark matter in this environment. What in the environment matters? And so the way we've decided to do this in my lab is to leverage this experimental oddity called lethal dose 50. And so here in the top left, we're using a model of sepsis that's commonly used. And the lethal dose 50 is basically where you take a whole bunch of animals. These are from the same litter, the same mom, the same cage, the same rack, the same room. So the environment is, for the large part, as controlled as one can make it. You give them a dose, and you can achieve these doses where half of the mice die. And what we think is going on and what we're testing is that, small changes in the environment, for example, not all mice ate at the same time last. Not all of them have the same kind of need to defend their body temperature. Not all of them experience the same kind of stress and so on. Things that we know clinically matter in inflammatory diseases, and then see if we normalize them, if we can shift the LD50 to an LD0 and LD100. And so here I'm giving you just an example. We were particularly interested in psychological stress, which we modeled in a couple of ways, which greatly enhances the response to this inflammatory challenge. And because Yale is such a beautiful place to do collaborative research with the cutting edge technology we have at our fingertips, I essentially run a small mouse hospital, and we try to measure as many things as we can, taking kind of an integrative physiology approach, and look at sophisticated ways of looking at metabolism, neurobiology, hemodynamics, et cetera. And then in reasonably efficient time, arrive at mechanistic dissection. And then with my clinical colleagues, in this case, the excellent departments in psychiatry and neurobiology, try to translate our basic biology discoveries in animal models to human samples. Next slide, please. And so we really have tried to apply this lens to studying coronavirus. My lab essentially has been working on this for the last couple of months. And we developed a model using a mouse beta coronavirus that belongs to the same clade as SARS-CoV-2, and which recapitulates a striking number of features of human COVID-19. And as you can see on the leftmost panel, unsurprisingly, if you give more virus, you can enhance lethality. But when you apply kind of our mouse hospital approach, we started noticing a couple of examples, which I'll show here. So for example, if you look at the oxygen saturation in moribund mice, not all of them are greatly hypoxemic. And if you take a group of them and look at, for example, cardiac damage, and we do this kind of in a multimodal way, as I showed you before, some of them have cardiac damage and some of them don't. And then, for example, if you take very old mice, so geriatric mice, you find that they're exquisitely sensitive to doses of coronavirus that are basically asymptomatic in younger mice. And so the questions that we're interested in are, we think that like with other systemic inflammatory diseases like lupus, for example, that it's probably one syndrome with many diseases, and that we are interested in what these endophenotypes are, what determines why you get them, and really to try to discover the unique biology within the endophenotypes. And we think that we can partner with the greater community here to try to apply kind of this lens to directing clinical therapeutics to the endophenotypes within the larger COVID-19 syndrome. Thanks.
Thank you, Andrew, very much. Ellen, if I could invite you to please turn on your video and audio and present your slides.
Sure. Hello, everyone. I'm really pleased to be here and be able to participate in Yale Engage. I think it's wonderful to come together with the broader community to see what we can do to fight this virus. And I'll tell you a little bit about my background. This is just a screenshot from my lab webpage, actually. Even before this virus came along, we're very interested in the respiratory tract and respiratory viruses. So what I'm going to tell you about is how we're kind of using that background to look in detail at this virus and what we might be able to do about it. So the micrograph that I show here on the slide is the lining of the nasal passages and conducting airways. So that's what the airways, the nasal passages and the large airways going down to the lung, this is what they look like. And what you're seeing is the epithelial cell layer. And these cells are really quite incredible in that they have a lot of natural antiviral functions. They secrete antimicrobial proteins, they secrete mucus, they have cilia, which you may or may not be able to see on the slide, but they have many different functions, and they also detect viral infection and signal and create antiviral responses right in sight, which can control viral infection at the very early stages. So next, please. So we think of the upper respiratory tract when the virus first enters the airway as the gatekeeper. And there's a lot of evidence that effective defenses in the upper respiratory tract keep pathogens out of the lungs. And a lot of times when you get lung infection, that's because that spillover enables pathogens, whether it be bacteria or viruses, to get down to the lungs. And so in my lab, we're interested in genetic and environmental factors that modulate the defenses at the very early stages where things could potentially be nipped in the bud or not. And that's going to determine the outcome. Another thing that can happen at these early stages is when that virus infects the cell and there's different signaling events, it can recruit different types of immune cells. When Dr. Iwasaki's mouse model slide showed different types of immune cells being recruited to the airway. And Dr. Wang mentioned how different environmental factors can really influence the course of disease, even in very similar hosts. So that's the kind of thing we're interested in. We think of the response at this early stage as like a marble sitting on top of a mountain. If you nudge the marble in one direction and it rolls down one way, you're going to get a very different outcome than if you nudge that marble in a different direction. So next slide, please. So specifically sort of focusing in now on how are we looking at this phenomenon as far as COVID-19? Well, as many of you are probably familiar with, the major way of testing for this virus at the moment is nasopharyngeal swab, which is shown here. This is actually on a child. And you can see that part of the airway that this swab samples is the part that's lined by these cells I just told you about. So next slide, please. Or just, yeah, there we go. So it turns out that when you do these swabs, you actually do pick up some of these respiratory epithelial cells that form the airway lining on the swab. And it turns out that you actually can look inside these cells and say, what is the pattern of genes that these cells are expressing? And even look and see what proteins and other mediators that you can measure that these cells are making. And they can, and we're looking at that in detail to try to learn the answers to some of these same types of questions that other people have mentioned, but sort of at a different stage of infection at that very early stage. And potential applications of this are, first of all, in diagnosis. There's been a lot of challenge ramping up testing to large scale or screening people at large scale to see who might be infectious. So biomarkers of replicating virus might be useful for that sort of thing. And that's one thing we're looking at. Also predictive biomarkers. If it turns out that certain gene expression or protein expression patterns correlate with which way that marble's rolling down the hill, that's something we could assess at an early stage, and then you know what kind of interventions you need. And of course, early stage interventions, all of these responses I'm talking about are not just biomarkers. This is the very early initiation of an immune response. So if you can understand which way do you want that immune response to go at the beginning, Maybe you could design interventions to make that happen more often in people. Or maybe you can find cofactors like other microorganisms in the respiratory tract, for example, that push that immune response in a good direction or a bad direction. So those are the kinds of things we're looking at. So yeah, so this is a figure probably people have seen different versions of this, and I'm sure it will continue to evolve. But there is this idea with COVID-19 that at the beginning, and it's not just an idea, there's data. At the beginning, this is an infection of the upper respiratory tract, which then progresses to an infection of the lungs, and then can even progress to immune-mediated pathology, which can lead to very serious illness. So where we're focusing on is stage 1 here. How can we understand these very early stages and nip this infection in the bud, learn how to do that? So the things that we're seeking partnerships for is, first of all, platforms to obtain more information from the clinical samples. So we're using the nasopharyngeal swabs as depicted on the slide, and we're interested in looking at everything we can to really understand what's going on. So if folks out there listening have a new platform for looking at characterizing some of these things I've talked about, that would be great to have a further conversation. And the other is we've already begun to identify some interesting biomarkers and would love to work with people who have are interested in point of care test development to use in ongoing studies to try to really make a dent in this disease. So thank you very much for your attention, and I'm happy to take any questions later.
Thank you, Ellen. Thank you. That was great. And so now we've heard just the beginning of the conversation from each of our speakers. I would ask you all to put your video cameras on, and we'll begin the discussion part of the morning. We have a few questions that folks have sent in advance, and we have questions that have come in through the chat. And I'd like to start with you, Akiko. So many people are interested in the possibility of a vaccine for this disease, and I know that this is something you're very knowledgeable about. Can you launch our discussion? And for the speakers, please join in the conversation as much as you like.
Thank you, Ruth. So this is a tough question to begin the discussion, but as you know, there are over 100 different vaccines that are currently being tested at various stages, some of them already in clinical trials. And vaccines are really the only safe way to get back to the society. And it's really, really important that we test the vaccines with, expedience and with care. The reason I say with care is that, the vaccine itself may not be have any toxicity. So the phase 1 clinical trials will show whether a vaccine is safe to use in people. But the phase 2 and phase 3 are also very important to determine whether a vaccine elicits robust immunity and whether that has no adverse effect on the infection with the SARS-CoV-2. So I say this because in the past, vaccines against SARS-CoV-1 was tried, and this unfortunately didn't progress through clinical trials phases 2 and 3 because SARS-CoV-1 was controlled already well because of public health measures. However, some of the preclinical studies that were done in primates and rodent models had shown some unintended consequence of the vaccine, such as antibody-dependent enhancement of disease. Now, this may or may not happen for this particular SARS-CoV-2, but we just have to be, careful, and we are. And the entire community is watching, and the people who are engaged in vaccine research are absolutely careful and critical about moving forward with their vaccines. But at the same time, we really need a safe and effective vaccine quickly. So I think the world is watching, and the world is hoping for a effective vaccine to come online. And we as a community of scientists can try to help look at these responses. And for instance, what are the correlates of protection? We don't know. We don't know what we're trying to achieve yet. Like, is it neutralizing antibody? If so, what is the titer that we need? Is it T-cell response? What else is needed? Is it mucosal immunity? So all these questions have to be worked out. But I know that for myself and others at Yale, we are very, very much focused on developing epidemiological studies to be able to look at these issues. The correlative protection is absolutely critical for evaluating vaccines. So, and there are many other things I can talk about, but I don't want to take too much time.
Well, Akiko, before we leave you, we just got a question in the chat, and it's right up your alley. People are really interested in the antagonizing of interferon activities. And this is something, for those of you who haven't read it, Akiko had a very recent review on all the interferons in viral infections, which is incredibly informative. But could you address whether the SARS-CoV-2 directly targets lymphocytes and antagonizes interferon activities? And then I promise the other speakers, we have questions for you as well.
Okay, so interferon is a double-edged sword. For most of the viruses, they have evolved mechanisms to antagonize interferon induction as well as interferon dependent effector mechanisms. So, you know, viruses can only succeed if they can evade or counteract some of these effects of interferon. And SARS-CoV-2, or I would say SARS-CoV-1 has been studied a lot more. And there are many viral genes that are known to block interferon induction and signaling, as well as some of the interferon stimulated gene functions. So, and we know that the viral sequences are you know, relatively preserved for some of these genes that counteract the host interferon response. And that's unfortunately also present in SARS-CoV-2. So given that the virus is very good at blocking interferon responsiveness, and the fact that we see, at least in our mouse model, that type 1 interferon appears to elicit recruitment of these inflammatory monocytes and other leukocytes into the lung, you know, first of all, the endogenous interferon is not very good at controlling the virus. And secondly, the interferon has this pathological effect. But I would be careful about interfering with interferon altogether, because we really rely on interferon to control the virus. But what may be a better strategy is to prophylactically or very early on treat with high dose of interferon. Not high dose, but like enough dose of interferon that we can block the viral replication. And that is a very different story from the endogenous source of interferon, which is already, we know, are being blocked by the virus. So we're getting like low level of interferon kind of late in disease, and that's not a good situation. Whereas if we can interfere with that kinetics by giving patients or people interferon very early on, that may be a good strategy. So again, these are all the things that we need to test in order to know if it's efficacy. Yeah.
Terrific. Thank you, Akiko. I'm moving to some of the other questions that are coming in from people listening to us today, and I'm turning to you, Ellen. There are questions about the size of the particles in the upper respiratory system, differences between upper and lower respiratory system in this disease. And I know that you're very involved in those elements of the immune response and the host response and in testing. And could you address some of those topics?
So the particle size, well, what's very interesting about respiratory viruses is, you know, is that in many cases, the infection starts in the upper respiratory tract and spreads down. And sometimes with bacterial infection, you just get aspiration. So it sort of skips and gets down to the lower lung and then can grow. But in almost every case that we did a study a couple of years ago where we looked to what extent do people with a lower respiratory viral infection have evidence of an antiviral response in the upper airway, and it was almost all of them. So, this virus seems to fit that pattern. So I'm not sure exactly if that's what meant by the particle size, or if you're happy to take any clarifying questions about that, but there seems to be a lot of good evidence that this virus starts with the replication in the upper respiratory tract and then travels down.
So Ellen, I think since our partners on the call today are interested in interventions, and perhaps one of the questions would be, is there some kind of microparticle size that might be used as an antagonist or a therapeutic to disrupt virus entry?
Disrupt virus entry. I feel like I don't know the answer to that. So if anyone else does, please feel free to chime in. I mean, our body naturally makes antibodies that are secreted into the respiratory secretions. That might be a great way, natural way of blocking viral entry. And hopefully that will be one of the things that one of the strategies long-term that, that results from a vaccine.
Terrific, thank you. And Naftali, a number of the things we've talked about have to do with the acute phases of the infection, but in your talk and in some of the questions, people are asking about long-term sequelae. I wonder if you could address some of those points.
So yeah, I'm happy to do it. What we know is that in general, if you have a severe lung condition like ARDS, the adult respiratory distress syndrome, which is actually the most severe manifestation of COVID-19, you take a long time to go back to functioning. So the post-ARDS syndrome is a big issue. Patients have mental, cognitive, and physical impairment, and they have some lung issues. Specifically with coronaviruses and the previous epidemics, we have seen some sustained lung damage and especially scarring and fibrosis. Now, this is not the progressive pulmonary fibrosis we see with idiopathic pulmonary fibrosis that people die within a few years. That's the disease of study. But there are, you can see fibrotic bands in CT scans even 10 or 15 years later, and these are really small and problematic series, but the scans are this. And the question is, will this happen in patients with COVID-19? We already are seeing the most common call we get now from these big victories, the patients that we've cured, the patients that you've heard the staff clapping to them when they leave the corridor. The biggest thing we see is that they call to the clinic and they're still coughing, they're still short of breath, they're still not well. And the truth is we don't necessarily know what's going on. And I think that this is an important thing. We've had so many people having the disease that even if 5 or 10% of them develop chronic sequelae, this is a major public health issue. And again, this disease, like many other diseases, hit the disadvantaged parts of our communities. So not only you have people coming from with lower socioeconomic determinants of health, now they're going back after the disease to the same relatively bad access to healthcare they've had. So this is really important. And the ability to basically fix it quickly so these people can go to back to work and life is really important.
And just before we leave the lung sequelae, Naftali, are there any observational studies or points about smoking which would be relevant? One of the people in our community was asking about that, whether nicotine might have a protective effect.
So I'll say two things. One is the data is not completely clear. Some reports suggested that smokers are less likely to be hospitalized, but others have reported that among hospitalized patients, actually the mortality among current smokers is higher. In patients with COPD, definitely the mortality is higher. So I would be very careful about saying that nicotine is protective. I want to highlight one thing, which is cigarette smoke and nicotine causes lung cancer, coronary heart disease, and numerous other diseases. And COVID-19 is critically important, but you don't want to cause yourself lung cancer because you try to prevent an infection.
I would say that's a good point. While we're on the subject of severity and predisposing conditions, Andrew, I wonder if we might turn to you with your expertise on immunometabolism. And for those of you who don't know, that old wives' tale, starve a cold, feed a fever, has now been published in Cell by Andrew Wang. So can you tell us some of the points about immunometabolism, which are perhaps areas where we could intervene in susceptibility to this illness?
Thanks, Ruth, for the question. I'll do my best to answer it. I think one theme that all of us are saying is that we're still learning a lot about this disease as it unfolds, and so these kind of research, clinical, pharmaceutical collaborations are all the more important. One thing that's I think, as you mentioned, very clear is that obesity, insulin resistance, and metabolic syndrome, underlying hypertension, et cetera, seem to be very strong susceptibility factors. And they share common features also with kind of extremes of age. And in the literature, it's called kind of meta-inflammation. And so one idea is that this meta-inflammatory state somehow primes the COVID-19 response. And so how that works is not entirely clear. And I think thinking about it from a clinical translation perspective, it's really kind of patient cohorts that you have to think about. So in whatever clinical therapy you want to apply, are you applying it to the right patient population that has the best chance of success? From that perspective, simple things I think we don't know a lot about. The study that you were referring to, at the heart of that question is a clinical question, which is, in these patients who are intubated and are fed, how should we feed them? And does the way we feed them, for example, change their outcome? And this question, not just in COVID-19, but generally is something that many of us are trying to understand. I don't know if I answered your question. I seem to ask more questions to your question, but I think you captured the gist of it.
I think asking the questions is what we're here at Yale to do, Andrew, so thank you for that. Let me turn to you, Carrie, because there are a number of points that rely on your expertise to help us understand. And we were first going with genomics, but I also want to address the issues of children, susceptibility of children, and points that you might be able to raise about the specificity of their diseases and susceptibility.
Great, thank you. Yes, I'll try to provide some insights, but certainly more questions than answers. I think one point that piggybacks off of some of the things that Naftali was just mentioning relates to the general population of primary immunodeficiency patients, because of course, there are different gene mutations, different types of immunodeficiency, but what do we know about that population of typically young people? And it's still very early days, so we don't have any really firm conclusions, but intriguingly, it doesn't appear that this population is that much more susceptible to COVID-19. So they seem more or less in the early data, and there are ongoing studies and surveys to try to address this further, but it's similar to the general population, meaning if they had a pre-existing lung condition, for example, so we see bronchiectasis and other lung damage occurring in kids and people who have CVID. So these are people who don't mount normal humoral immune responses and therefore have recurrent sinopulmonary infections historically in the past before COVID. That can cause lung damage. And now those people do poorly with COVID-19, but generally, if they don't have that pre-existing condition, we're not seeing a tremendous increase in susceptibility. So I think we need to do more to tease that apart and really understand, as Akiko was mentioning, what are the clinical correlates, and do we see that they mount normal antibody responses, normal T-cell responses? What can we learn from that? So I think thinking about that population as a whole is really interesting. And then turning to the MIS-C children, the ones with this no prior history, who now have this inflammatory phenotype. Again, a lot to learn there. And I think one of the things, I've seen one of the questions there in the panel, in the chat, that they're wondering if there's some correlation between neutralizing anti-SARS-CoV-2 antibodies and what those antibodies see relating to the pathology of this inflammation. And those are perfect questions and things that if there are folks in the audience who have great ways to assay for this, that's a main question that we're looking at, is what do antibodies in these kids recognize? Both which parts of the virus, so trying to understand if there are certain antibodies made against the virus that then cross-react with self. So these are not known yet, but important questions to ask.
Thank you, Carrie. And before we leave you, can you just address thoughts about vaccines in children and whether we might adopt the use of a vaccine in children as a population rather than elderly?
Right, I think it's an important question. I mean, in early days of COVID, the thought was perhaps that population doesn't, it's not as urgent to have vaccination in that population because they weren't as severely affected with the respiratory presentation. But now I think it's becoming clear that there are other manifestations of these infections. So what we know, again, going back to the 4 to 6 week delay, and it's still very early days, but one of the things that I'm thinking a lot about is, well, okay, so let's say they were exposed to SARS-CoV-2 4 to 6 weeks ago. Now they're showing up today in the PICU. Why? What triggered that? And as Andrew points out, is there some environmental trigger? Is it a re-exposure to SARS-CoV-2? Is it an exposure to a different coronavirus? Kawasaki is thought to be triggered. This is a similar disease thought to be triggered by some kind of viral trigger that hasn't been perfectly well defined, but there are suggestions that other coronaviruses could do this. So I think a lot of these answers, thinking about should we vaccinate children, I think yes, if we can build evidence to suggest that it can be done safely. So if it is true that you mount a response and then it's re-exposure to some second trigger that makes them ill, if we can prevent that first exposure and modulate the immune response prior to them developing these potentially pathogenic adaptive immune responses, that would be ideal.
Thank you, Carrie. That's great. And in response to one of the questions from the chat, I would refer you all to our new website at Yale. It's covid.yale.edu. That reflects clinical trials in progress, investigators at Yale who have a specialty interest in COVID, publications from Yale authors, which are updated every 24 hours, preprints and published papers. So that's a very good resource. So there's a couple of general questions that appear regularly, and would like to throw this open to all of our panelists. Two kind of related points. One having to do with inflammation, which we all study. Is it good or bad? Obviously, that's complex. And the second question, which several people have asked, what resources would be the best advantages for what we need to do? What are the constraints that are holding us back in these investigations? And just raise your hand or jump in, whoever would like to address some of those points. Naftali.
So I can take the lead. You know, listening actually to the speakers and actually looking at the literature, I think that one of the strongest points for industry, academia, collaboration is actually teasing out this sort of this double-edged sword that is inflammation in COVID-19, because we have models, we have the patients, we have a lot of the science, but we don't have the access to all the modifiers. And many of the companies, startups, big companies have actually libraries of immune modifiers. And in theory, if we could do a really big like COVID-19 Connectome project in which we actually study all these effects of all these molecules, while allowing the individual scientists, of course, do their own scientists, but also generating information that is useful for everybody. We may be able to start calibrating what is bad, what is good. Because for me, like I immediately tweeted Dr. Iwasaki's interfering with interferon because I loved it, you know. And the same thing, so don't interfere with interferon and probably not with IL-1 beta. And we're interfering with IL-6 all the time now, right? So what does it do? So I think we need to think about it. I'll let other people chime in, and then I may something about resources afterwards.
I can, my first reaction when I heard is inflammation good or bad? Of course, the answer is both, because these immune responses are how our body fights infection. I mean, they're very, very important. If you got rid of them, we would not make it through the first 5 years of life, you know? So these are very important. The immune system has got a very important role to play, and we don't want to completely block it. So the key is to understand, you know, as many of the speakers referred to, what's the path you want to sort of push the immune response down, and which one don't you want to go? And as several speakers also alluded to, this really depends on the timing. So very early in infection, you know, that interferon response in the upper respiratory tract is incredibly effective at blocking viral replication. But later on, you know, as Akiko was referring to, that could have some detrimental effects down the road, depending where you are in the process. So I think it's not all good or all bad. We're right. It's both, and we just really need to know the details. You can't make that's what we're all about, is let's get into the nitty gritty. And as far as resources for the kind of work like that we're doing and several other people, and we talked about the biorepository, I think technology that allows us to get more data out of these samples is incredible. We don't know what's going to be the most important thing. So technology to use a small volume of sample and broadly survey for different types of molecules, very useful. And personally, I think not waiting until something is established in 2 or 3 years to make rapid tests that we could potentially bring out in the community and start using and gathering the data in real time. If people have a novel platform for that, I mean, personally, and I'm sure others as well, would be very interested in collaborating to highlight the new technology and how it can help the community. So that's my 2 cents on that.
Yeah, just picking up on what Naftali and Ellen said, I think Yale is uniquely positioned to really push forward this question about inflammation. And it would be an ideal time to think about building an institute around this question, because inflammation, even though it has a terrible sort of reputation in the public, it's actually, as Ellen said, it's critical for fighting infection, and not only that, just to sort of the physiology of human beings in general. For instance, inflammation is what keeps us, alert and learn new things. And, neuronal pruning, for example, is mediated by components of the inflammatory system. And lots of things that we still don't understand, what Andrew Wang is studying, like how metabolism interface with the inflammatory system to allow us to fight infection, but also determine the course of infection, what happens once we get the infection. So this is actually a prime time to start thinking about a much bigger picture than just COVID-19, to be able to deal with the inflammation as a biology in general.
Akiko, I was going to say almost exactly the same thing you were going to say.
Sorry.
No, it's perfect, because my perspective is very similar, which is that we're just starting to learn all the ways the immune system is involved generally in human physiology. And from a clinical perspective, it's important because, depending on how you look at your intervention and what you want to get out of it, if you're kind of very myopic about what you think the response should be, you miss a lot of things. And so, as Naftali brought out with IL-6 therapy, the rheumatologists who used tocilizumab learned later that tocilizumab has this off-target effect in its effect on lipid metabolism in a way that it's unclear if it's beneficial in the long term for patients with inflammatory diseases. And so I think having a broader perspective on how immune cells interact with other systems, not just with infection, but in human health in general, is really important, especially as we're thinking about making these interventions in a large number of people.
And I'll just chime in to say I completely agree with all of those points. That hits the major constraints with Ellen in particular. The doing more with less sample for pediatric kids is that much more important. It's always important, but we don't get massive amounts of blood from these kids. So technologies where with a few microliters of serum, we can look at all these biomarkers, do some antibody assays is good. And I think also thinking about the age spectrum. So being able to do things in the very young and the very old. Again, they're still harder to identify and harder to analyze, but this idea of, on the other side of the spectrum, older folks, so for example, in nursing homes that are resistant to infection, and thinking about ways in which we could have coordinated efforts, ideally in something like an inflammation institute, as Akiko points out. And I'll also add different ethnicities and of course socioeconomic status, and how do we make sure that we're studying across the whole spectrum to glean the most information genetically and also with regards to inflammation.
Yeah, and I would chime in just to echo what Carrie said. These different age groups that naturally differ in their immune system. Like the immune system of a 5-year-old child is not the same as a 20-year-old and is not the same as a 65-year-old. And it's sort of a natural setting in which we can understand differences in immune responses and outcomes. And this disease is like for kids, it's very striking. I mean, on the one hand, it's interesting to study these rare examples of severe illness. But on the other hand, it's also interesting to study what is it that makes these children not in general, not highly susceptible to pulmonary illness. There are exceptions, of course, but that's sort of something that could help the adults if we can understand how that happens. So I think there's a lot of opportunity, this disease and this fight that we're in against SARS-CoV-2 really highlights it.
And to add to that, I think also intersecting with Andrew's interests, one of the things that is striking as we're continuing to recruit pediatric patients, those who manifest with the severe respiratory phenotype, the children, are almost always obese. So again, that layer of that additional variable and how an otherwise protected population can become susceptible with certain environmental or external factors.
And teenagers and older as well, like in general, as opposed to the really young kids.
So So again, and I saw some questions and that got me thinking. So the most dangerous thing for science, exactly like for nations, is to fight the previous war. And I do think we'll have a vaccine for COVID-19. It will take a year or less or more. We'll probably have some effective therapies. But there's going to be other things, right? No matter what, we sort of disrupted global boundaries. We move around. All the environmental stuff our governments are not doing anything about. There's going to be additional disasters. And it's almost like our role as citizens is to act against them and politically, but our role as scientists is to find ways to deal with them in some ways. And the question is, and that's why I like, again, Professor Iwasaki's and everybody else's comments, is thinking about using regulation of inflammation, injury, and repair to actually deal with the next pathogen or the next noxious agents. I got interviewed yesterday about how do you protect yourself from tear gas. Like, I would never have expected to be asked about this, in the US. So thinking about these things and utilizing mucosal immunology and other things to really protect and enhance ourselves is, I think, would be exciting. And this could be a place, again, academia and industry and government come together in one place, an institute would be really exciting and useful.
Hi, this is Kathy Lynch. I have a question for our panel as well, and picking up on something, Naftali, that you said a few moments ago. As we think about what we've learned from these few months and the collaborative science that you've been describing and the collaborative efforts and interdisciplinary work and even our partnerships with industry, what of these learnings would you like to move forward and hold on to? How can we continue this collaborative focus to fight not only COVID, but the future issues and diseases that will come our way?
Maybe I can jumpstart this. I mean, I think as people are saying, one common feature of many chronic diseases that are the kind of the current epidemics of our time involve inflammation. And it's unclear if, as Akiko and Carrie and other people have talked about, is that immune response that we're measuring, is that good, trying to fix some problem, or is it bad in driving the problem? Or as Ellen said, is it both? And we just have to know kind of how to toggle it. I think that, as horrible as a global situation this is, it can also be a unifying one to move forward to help think about these ongoing diseases that have inflammatory components and apply this kind of collaboration to those efforts. Not just, infectious diseases, but as Naftali was saying, I'll take that as a cue that I'm done.
No, Andrew, please finish. That was a technical issue. Please go ahead and finish what you were just saying. I think we didn't hear you.
No, I was just joking. I was just gonna kind of springboard the conversation.
Yeah.
I could also.
Oh, go ahead, Carrie.
I was just going to say, also add that I think Yale and our partners should continue to invest in the kinds of collaborations we've already set up. So building on the toggling, so inflammation is both good and bad. We're making great progress in trying to in defining the things that determine that. So for my side in the rare disease world, there's a new entity beyond primary immunodeficiencies. There's a more recent entity of autoinflammatory diseases. So as Andrew knows well, these are monogenic defects in kids who don't really present with more infection, at least not infections that can be defined, but instead just have this high, sort of chronically high inflammatory tone that is often due to mutations in things that we have evolved genes that are really important to control that inflammation. And as we continue to build these data through the kinds of collaborations we talked about and working broadly in mouse models and in humans, I think we're going to define ways that we could therapeutically adjust the rheostat and sort of, in this context, this patient, this disease would benefit from more inflammation in a higher, maybe earlier triggering of these events, whereas other diseases we need to tune down the inflammation. So continuing to invest in really the fundamental basic science, also applied directly in humans, is critical.
One of the things that was really to some extent thrilling during the surge was actually the solidarity, right? So people really enrolled to help, right? We didn't break rules, but we broke boundaries. And I think these broken boundaries have generated, even in Yale, we all know each other, but we all sort of left our molecule of interest and sort of mobilized to do something different. I don't know how you can maintain this spirit for a long time, but if we can, and potentially that's by the support of industry and government of creating these extra siloed spaces that you could actually work together dealing with one question, with one global question would be exciting. The other thing I want to highlight, and that's really, I think, unique to Yale and New Haven. So it's Yale School of Medicine and Yale New Haven Hospital. And we take care of everybody as physicians. There's no safety net hospitals or boutique hospitals in New Haven. And I think that's an important thing because it allowed us both to give the best care to everybody in our community, but also it allowed us to do research on all the people that usually our research communities are blind to, like underrepresented minorities. So I think there is a huge opportunity here to continue this spirit. If you look at the numbers at Yale because Yale New Haven Hospital protocolized therapy, everybody got relatively the same therapy. So some of the social determinants of care, like implicit bias and others, were less active because if a patient had 3 liters of oxygen, that's a drug they got. So I think if we could continue these things, then we could really be transformative, and especially in the study of inflammation and environmental injuries. So that's what, if we could even maintain a little bit of our spirit of outside of the silo and of scientific inclusion would be really exciting.
Did you want to go?
Oh, I did. I was letting you go.
Oh, okay.
Go ahead.
Okay, yeah. I mean, in terms of thinking about this engagement with the industry, one of the things that really, and in thinking about future pandemics, as Naftali was alluding to, we really need great set of antivirals. We don't have the arsenal to fight most viral infections. And there are lots of great antivirals that were developed for HIV and hepatitis virus and so on. And that's wonderful. And even to some extent, Ebola. But we need a basically, arsenal of all different antivirals that work on this class of virus and that class of virus. And, I feel like that's missing. And I know that lots of companies are working on that, but if we can use this as an opportunity to really mobilize that effort in a very unified and global manner, that will serve the humanity well in the future years to come. Because I feel like if we had a Tamiflu equivalent to ready to go for this virus, it would have really changed the course of transmission and disease. So that's another thing that I think one of the learnings, Kathy, I got from this is we really need great antivirals.
Actually, the point I was going to make just dovetails right off of that, which is please, 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 let's be ready for the next pandemic. We need, if this really highlighted economic disruption and the health consequences, we know now, let's not forget that this can happen. And so not the antivirals, that's a great point, as well as let's be looking for these viruses before they're spreading around the world. And there's a lot of tools that people are developing in response to this pandemic that do apply going forward, and concerted effort rather than playing catch up, like a perspective effort rather than playing catch up would be something really, really worthwhile and beneficial. And as Akiko was saying, you know, there are classes of viruses. Even though this virus is novel, it has a lot of similarities to other viruses. So just getting the broader knowledge on how to fight these different types of viruses, it really does translate over that you can just jump right in when there's a new virus and figure out what will probably work and really get a shortcut. So I really want to put in a plug for just thinking about how we can broadly and systematically be ready for this.
And Ellen, that raises also the next 2 workshops in the Yale Engage series, which are going to be addressing issues more in the public health realm and the community points that Naftali was raising. Andrew, you raising your hand?
Oh no, I dropped my discipline of touching my face for a second.
Ruth, I did see a question in the chat. Some people are bringing up how school is reopening soon and wondering about thoughts about that and in particular the pediatric presentations. So I can tell you what we know in terms of the inflammatory syndrome ages. It's older than in Kawasaki. So Kawasaki is usually 2 years or younger. The criteria are generally 5 years and younger, but most of those end up being 2 or younger. For this Miss C presentation, it's a bit older. So you see kids anywhere up to the teens. So low 20s, we've seen them with this kind of an inflammatory phenotype. So yeah, it's definitely would be affecting school children, but it's something, as we pointed out, we need to study further. I should also say most children recover. So it is a sort of temporary inflammatory, very severe inflammatory response. There is the potential complication of coronary artery disease, as is seen in Kawasaki. But most kids are treated with anti-inflammatory things, including IVIG, sometimes IL-6 blockers, sometimes IL-1 blockers, and steroids, and they tend to bounce back. So the kinds of things that show up on these kids, I think that was asked, what would you watch for, is fever, the persistent fever, and this rash, abdominal pain, and those are the kinds of things that typically present.
And of course, as immunologists, it's really fascinating why the organ systems would be affected so differently in hosts of different ages. But there's a lot of stuff we don't understand, and the basic research just needs to be done. And it highlights some of the things we've talked about also, that the mouse model is very valuable, but human investigation is very valuable. And one of our themes today is translational work, which everyone on the panel is involved in, which presents opportunities and challenges, of course. Panelists, do you have a few more thoughts about anything that's constrained in your work or new directions we haven't covered up to this point?
I saw a few questions about protecting the lung, and I'll just mention again one of our challenges, and we've heard today about 2 models of COVID-19 in mice. I think it's critically for us to have models of actually COVID-19 injury on human tissue, either by organoids or by the way we do it, precision cut lung slices. But again, even if we have the model, the university lab can look at 1 or 2 or 3 targets, right? So it's really would be exciting if we could think about what I said for COVID-19, the COVID-19 Connectome project or something like this, in which we can very quickly screen a lot of compounds. So we could, if we did that, we could screen the nasopharynx and prevent infection. We could screen the upper airway and affect bronchopneumonia. We could screen the lower lung and potentially affect ARDS. Some people have asked, I think there was one question about surfactant therapy. The injury to the lung is complex in COVID-19. It's what we call both intrinsic, so directly from the virus, and extrinsic from the inflammatory response, both hitting the epithelial lining and the endothelium. So it's a little bit more, it will require some thinking and potentially even combination therapies or things that change the metabolic state of these cells. But if there was a way to start thinking about screening a large libraries of compounds with their lung protective effects, from my point of view, would be really exciting. And I'm sure for the immune modulation effects for other people in this call.
And Naftali, are any of the newer models of lung tissue I know that are active in your section, because our labs are on the same floor, for instance, like the organoids and some of those elements, would those be valuable in vitro testing?
I think so. I think human precision cut lung slicing is going to be very important. The question is, what we do is we take normal human lungs rejected for transplant. Actually, now there is a problem because they need to be tested for COVID-19. So actually our supply hasn't, and the idea is you cut them, you can keep them in culture for, 7-8 days, actually more, and you can treat them. So in models of fibrosis, this is very accepted. In models of infection, people have done less with it. Of course, it misses the effect of circulating immune cells, but you can still learn a lot. And if you do it on nasopharynx airway in this, and we can envision also more complex model, you can envision, humanized immune mice and other things. So, but I think that an investment in this early, not waiting for the NIH life cycle. So you submit a grant today and get funded in a year is really important. And then screening beyond your sort of myopic or my myopic hypothesis, but bringing in the sort of the power of pharma would be really helpful.
Right, and one of the points you made a few minutes ago, Naftali, about how the urgency of COVID-19 has brought all of us together in a kind of a shared mission and an unbelievable pace. And of course, the preprint servers, which have really come to the fore in this spring, where we are able to share with other scientists advances, hot off the presses, even though you have to understand they haven't yet been through peer review. And we have several in our panel who are also masters of Twitter. So you can follow Naftali and Akiko and learn a lot of science on a daily basis. We're closing up near to the end of our hour. We can hear a few remarks from Mike Crair, and I wanted to remind the panelists, the attendees, that you can continue to send questions to Kathy Lynch. We have 2 more Engage sessions planned with Yale faculty on different targets, as you see on the slides now, surveillance and modeling, and more policy and economic topics coming down the road. But I hope, Mike, that you might have a few comments to make or any questions that are lingering. We have a few minutes left, but we are winding down.
Sure, Ruth. Thank you very much. It's an amazing discussion today. I just want to thank our Yale faculty for their devotion and dedication to discovery and to translating their discovery to success in treating and preventing diseases in the future. I also want to once again invite collaboration with our colleagues across the country, around the world, industry collaborations, other academics. We're in this for the long run, and we are really fighting a devastating disease, but we are firmly committed and invested to really conquering the disorder. So if you have any further questions, don't hesitate to reach out to me, any of the panelists, to Ruth, to Yale, and we'd love to see more of you and invite further collaboration.
Thank you, Mike. Thank you. That really says it all to everybody. And in answer to some of the questions, yes, the recordings and the slides will be available. Kathy Lynch, whose email address is here on the screen, would be happy to help you target questions to particular people. There were a lot of questions. We have a very lively audience today. We tried to get to as many as possible, but we may have missed a few. And with that, I think we can thank everyone for their attention and look forward to seeing you in the future series.
Thank you.
Thank you.
Thank you.
Thank you.